What is selective reporting? How does it affect research reliability? Definition of selective reporting. Selective reporting, also known as outcome reporting bias, publication bias, or colloquially as cherry picking, is the practice of presenting only those results that support one's hypothesis, while suppressing non-significant, negative, or contradictory findings. It is a form of scientific misconduct that distorts the evidence base and misleads readers. Selective reporting can take several forms: reporting only statistically significant p-values, p 0.05, while omitting those that are not significant, changing the primary outcome after data analysis. A practice called harking hypothesizing. After results are known, omitting adverse effects in clinical trials, and failing to publish entire studies that yield null or negative results. The file drawer problem. Examples of selective reporting. A clinical trial measures 10 different outcomes. For example, blood pressure, weight, mood, sleep quality. Only the 3 that show statistically significant improvement are reported in the published paper. The other 7 are omitted. Readers are left with an overly optimistic picture. A researcher collects data from 200 participants. The original hypothesis is that variable X predicts variable Y. After analysis, the correlation is weak and non-significant, p equals sign 0.30. The researcher then tests a different relationship X with Z that does reach significance, p equals sign 0.02, and reports that as if it were the primary hypothesis. This is harking. A study finds that a new drug has no effect compared to placebo. The researchers never submit the paper for publication, and the results languish in a drawer. This contributes to the file drawer problem, where only positive studies are visible. How selective reporting affects research reliability. Selective reporting undermines research reliability in several profound ways. Distortion of the evidence base when only positive results are published. The scientific literature becomes systematically biased. A meta-analysis that synthesizes only published studies will overestimate treatment effects and underestimate harms. For example, the antidepressant reboxetine was approved based on published trials that selectively reported positive outcomes. A later meta-analysis that included unpublished data found reboxetine to be ineffective and potentially harmful. Wasted resources. Other researchers may pursue false leads based on an incomplete literature. They may design studies to test a relationship that actually does not exist, wasting time, money, and participants. Irreproducible findings are a direct consequence of selective reporting. Erosion of public trust. When patients, policymakers, and the general public learn that negative results were hidden, they lose confidence in science. High-profile cases. For example, the selective reporting of antidepressant trials have damaged the reputation of medical research. Ethical violations in clinical research. Failing to report adverse effects can harm patients. It violates the ethical principle of beneficence, maximizing benefits and minimizing harms, because patients are exposed to risks without full knowledge. Reproducibility crisis. Selective reporting contributes to the replication crisis in psychology, medicine, and other fields. Attempts to replicate published findings often fail because the original findings were capitalized on chance or selectively reported. Prevention strategies to counter selective reporting. Researchers and journals have adopted several measures. Pre-registration of studies. Researchers specify their hypotheses, primary outcomes, and analysis plans before data collection. for example on clinicaltrials.gov or the Open Science Framework. Any deviation from the registered plan must be justified and reported transparently. Reporting guidelines tools such as Consort for randomized trials, Prisma for systematic reviews, and Strobe for observational studies require authors to report all outcomes, including non-significant ones. Mandatory publication of all results. Some journals and funders require that all results, positive and negative, be made available, either as a full paper or as a brief report. Data sharing raw data allows others to reanalyze and detect selective reporting. In conclusion, selective reporting is a serious threat to research reliability. It is not merely a minor infraction, but a form of misconduct that corrupts the scientific record, misleads clinicians and policymakers, and wastes resources. The movement toward open science, pre-registration, and transparent reporting is the only sustainable solution.